Welcome everybody to Herpetology Hangouts hosted by the Canadian Herpetological Society. My name is Pamela Rutherford. I'm the president of CHS. And before we get started with today's talk, I just want to let folks know that our next speaker in our series is Mike Mcfadden, who will be joining us from Taronga Zoo in Australia. The talk will be on Wednesday, May 14th at 8 p.m. Eastern. And Mike will be speaking to us about ex situ conservation programs for reptiles and amphibians at the Taronga Zoo in Sydney. I'm going to turn the introduction of today's speaker over to Julia Riley.
Welcome, everyone. Hi, so I'm Julia Riley, and if folks don't know me, I'm an NSERC postdoctoral fellow hosted at Dalhousie University. And just before we begin, I would like to share a bit about myself, my heritage, and recognize the ancestral history of Canada. So I grew up in Ontario, spying salamanders and garter snakes around now in forests and flowing rivers that surrounded my home. My parents are Kathy Lindsay and John Riley, and my ancestors emigrated to Canada from Europe. I want to pay my respects to the Wendat, the Haudenosaunee, and the Anishinaabe peoples, who are the first keepers of where I grew up as a child. Today, our speaker and I are coming to you from the Western Cape of South Africa, that are the traditional lands of the Khoikhoi and the San. I also want to express my deep gratitude to those who have guided me to see nature from a different perspective than my own. Thank you, Elder Tony Shigano from the Chippewas of Nawash, as well as Anthony LaForge, Kimberly Charles, and Elders Joe Naganash and Roger Jacqueline from Magnetalon First Nation. Team Miigwech. So today we are meeting virtually coast to coast across Canada, and I would like to acknowledge the ancestral and unceded territory of all the Inuit, Métis, and First Nations people that call this land home. As herpetologists, our natural lands and the animals within them are so important to us, and this recognition is a renewal of our commitment as a society to listening to and cherishing the knowledge that Indigenous people have of Canada's reptiles and amphibians. So greetings to everyone here today and to your ancestors too, wherever you are. So today we have a presentation that I am very excited about from Dr. Robin Maritz, who's a research affiliate at the University of the Western Cape in the Department of Biodiversity and Conservation Biology. And without any further ado, I think we can turn it on over, and I can't wait to see the talk.
Excellent. So thanks again, Julia, for that introduction, and thank you everyone for the invitation to speak to you all. So my current research addresses what snakes eat and why they eat those things. And so specifically, I'm interested in understanding the processes that shape the diets of snakes and the functional roles of snakes in African ecosystems. And the focus of today's talk will be to address how social media can be used as a community science platform to gather data on difficult to detect ecological processes like snakes feeding. And essentially, now that everyone has a camera in their pocket in the form of a cell phone, researchers have the assistance of potentially thousands of people who can act as community scientists. And so community, or you may know it as citizen science. They are volunteers who contribute to the collection and processing of data. And there's a long precedence for including volunteers in biological research. And it's really been the field of ornithology that has arguably been the most influential and the best at this. So they started back in the 1900s, early 1900s, doing the Audubon Christmas bird count that continues to this day. But there are also feeder watch that started in the 1970s, which actually originated in Ontario and continues to this day. And then with the advance of technology, there's platforms like eBird who, they're contributing thousands of observations every single day. And so you need to tap into what the ornithology community is doing and move it into the herp community. And so, there are many forms of community science. I've just alluded to a few of them there. And there are projects that are drawing on a range of community scientists that they have varying interests, whether it's birds or herps or plants, whatever it may be. And there's, they all involve various commitments from both the researchers and the community scientists. And so projects can range from very hands-on, so you have like a field assistant that's helping you collect data, to something that's very hands-off, where people are just engaging with photos and there's very little hands-on time that you have. But I want to highlight that because there are many herp-specific challenges, drawing on the observational power of community scientists can really bring huge benefits to a research project. And so for one, I'm sure many of you have experience with issues around detectability, especially if you work on snakes. A major benefit of community science is simply you have more people, there are going to be more detections. And similarly, adding more participants allows for a greater geographic area to be sampled. And so even if the study is located at one small site, you still have more eyes on the ground. And so 10 people versus 2 is a much larger search radius. And then with the addition of a global network, the geographic area sampled can be much larger. And the other benefit is that even if people fade out of the project, or people move on to other things, you still have this growing data set because more people come into the project, and you build this long-term data set that can continue accruing observations that can be used in monitoring projects. And then finally, for herps, which often have a bad reputation, community science projects can be hugely beneficial for what are highly persecuted taxa or seemingly low conservation value. Okay. And so in the following few slides, I just want to walk through a few examples of how community science has been used and how community scientists have really contributed to projects. And if you're at JMIH in 2019, back when we could actually meet face-to-face, there was a symposium dedicated to citizen science and herpetology. And one of the organizers being Russell Burke, so the research I'm showing here. This study took on a traditional approach where a bunch of volunteers came and joined a survey. And these volunteers contributed data on nesting predation of diamondback terrapins, and also where they're nesting, and also depredation. And so just having more people in the field really facilitated that project. And another important way that community scientists have contributed to herpetology is through longstanding natural history and geographic distribution notes from Herp Review, which I'm sure many of you are familiar with, or other society bulletins. And it's these ad hoc encounters and observations that often provide critical insights into the biology of species, and they can really be the foundation for future work. And so other ways that ad hoc encounters and ad hoc data have been useful is through platforms like iNaturalist. So with the advent of some of these technologies, we now have a growing database of occurrence data. So HerpMapper is another one that's big in the United States and Canada. But here we also have some local projects like Reptile Map. And I'm just showing an example of a study that combined these data to say, we have this species that has now pushed its range, probably speculatively due to the movement of plants, but it's just a good example of how long-term data can contribute to understanding how species are moving around. And so there's also an interesting way that you can use these data beyond just distribution, understanding the distribution of species. This paper just came out in February from Brianna and Greg Pauly, and I highly encourage you to go take a look at this paper. They used observations from iNaturalist and alligator lizards to test for differences in predation risk between urban and non-urban habitats. So it's a really clever way to make use of all these data that are available that goes beyond just making distribution maps. And occasionally you get some very dedicated people who are willing to contribute directly and independently to a project. The study that is shown here, Grandmothers and Deadly Snakes, an Unusual Project in Citizen Science, There were quote unquote fantastic grandmothers who contributed a number of observations of Hydrophis, and they show that without them, they would have never been able to collect as much data as they did. And so again, emphasizing how getting more people involved can improve the amount of data collected. And some of you might be familiar with Frogwatch, and it's an interesting case where it began quite small scaled in small chapters where you would get trained to identify frog calls and then conduct surveys at either an identified reserve or locally in your backyard. But it's something that is now moved to, you can record it with your phone, and you integrate the technology along with somebody who verifies the IDs. And so with technology, these monitoring projects have really advanced. And lastly, I want to highlight in terms of these field sampling events, this project out of Taiwan, the Taiwan Roadkill Observation Network project, which has had huge success raising awareness about their project, and participants are encouraged to place a scale card next to any dead vertebrates that they find on the road and submit pictures to the app that was created. And additionally, if they want to, it's hard to encourage sometimes, but people can also send the sample to the researchers. And you must take a look at this paper. The numbers are absolutely striking. Christina found with only 3,400 participants, they collected 45,000 road-killed items in 6 years. And the majority of those were snakes, which are accrued to around 16,000 specimens. So again, it's another example where even with a few people, 3,000 is a little bit more than a few, but you can really create a huge amount of data. And then finally, what I'm calling the data processing stream, there are several examples of that in herpetology. So if any of you have ever been on iNaturalist, anytime you confirm an ID, you are contributing to, you're contributing to processing of data. These species IDs can be useful to get it to a research grade, which helps when people download those data, but they're also useful because it helps feed back into the machine learning process that iNaturalist has in terms of identifying species. And then similarly, Andrew Dosso and his collaborators at University of Geneva have been working on this exact problem where how can you get a computer to identify a snake? And it's of particular interest when it comes to regions that have a lot of snakebite issues. And so by asking people to go and identify these snakes, you're contributing to the machine learning process. And finally, I've included these last two snapshots for, not because I have found anything directly related to herpetology, but these platforms really are potential that we should look into. These platforms offer participants the chance to go through and identify images from camera trap data. And they've been really important for digitizing museum specimens, including even the transcribing of old field notes, which I think is amazing. So this was just a brief overview of some of the ways that community scientists have contributed to herpetology, and I hope highlights some of the potential approaches that can really help facilitate more data and the efficiency of collecting data. But as I mentioned in the beginning, I'm interested in snake feeding ecology, and in case you aren't aware, dive studies would be really easy if we could just go out into the field and simply find them feeding, but these events are really quite rare. They're temporally and spatially unpredictable for most species. So historically, the way that we identify what snakes eat is it's useful to have ad hoc observations. This is, as I mentioned, the natural history notes in the Herp Review. And these are when you occasionally find a snake, you force it to regurgitate its prey or collect it for dissection. But this is very invasive, right? And time consuming and often produces low sample sizes. And so as a result, the adopted method has been to dissect museum specimens, which again is powerful, but you're limited to whatever is available at the time in the collection you're working in. And there's also important biases that we need to accept. And so if you work on ambush predators, like I'm depicting in the top right there, like Rulon Clark with the rattlesnakes in the US, or Xavier Gladys, who worked on puff adders here in South Africa, you can place a camera trap on animals that are in ambush, and you can record their behavior in terms of feeding or what they're eating. And finally, I put this photo here. These are obviously avian reptiles, but I just want to highlight how photo sampling has proven really to be a powerful tool for collecting dietary information at a really unprecedented rate. And as you may have guessed, the final approach, which I will focus on for the remainder of my talk, involved using social media as a community science platform to collect data on the feeding ecology of snakes in Southern Africa. And so the idea for this project really came about because while flipping through Facebook, there's literally, it wasn't uncommon just to see, for instance, a boomslang taking a bullfrog or a brown water snake with a mouthful of a ghost frog. And there are even photos of things like rhombic egg eater, which is quite a specialized feeder. They only eat bird eggs, or even very well camouflaged species like this gaboon adder. And so we're flipping through, we see all these, and then we also encounter photos like this ground hornbill, which is eating a puff adder. Similarly, things eating mole snakes, and even people were posting photos of the smaller herps, so geckos eating moths and rain frogs eating some alates. So after seeing these photos scattered across Facebook and recognizing their ecological importance, my collaborator and partner Brian Maritz, along with Andre Kutzer and Luc Verboek, created a Facebook group in 2015 dedicated to collecting these predation events in sub-Saharan Africa. And the rationale was that these observations, they're being posted regularly on snake groups or various photography groups, and that these should be gathered in one place and eventually turned into data that could be useful ecological data. And so Facebook needs no introduction. Obviously, it's well known at this point, but I just want to highlight how it's so powerful because of the fact that there are tens of thousands of users in a given geographic area, and they're logging on a regular basis. And importantly, we're just piggybacking on people's desire to share interesting things that they've seen without any additional login requirements. And it's also about, it's a social network, right? So it's about a network of people who can also share observations from a number of other groups who can then act as scouts for our projects. So we request that people share a few pieces of information when they post or share a photo, including an ID of the prey and the predator if they know it, but importantly, the location, date, and time of the observation. And then there are several pieces of information that I record from the photograph, including microhabitat, life stage of the individuals involved, an ingestion direction if it's a snake predator, and making sure to save the permalink as well as the photos and the videos. So how effective have we been? So to date, we've received over 2,000 feeding records, and half of those have been snake feeding records. So almost over 1,000 records now of just snakes eating. And notably, 75% of them come from South Africa, so there is a bit of a geographic bias, but they do extend up into Africa. And also the observations received are somewhat tied to where people are. You can see that the observations are hugging the coast there. Okay, so what I'm depicting now is, with these waffle plots, is the diversity of prey observed in the diets of the reptiles and amphibians, and similarly, the diversity of predators feeding upon herps in our data set. And so, yeah, roughly 129 families of prey in our data set and 89 families of predators in our data set. And I just want to highlight some contributor insights. And I think this really speaks to why we need to draw on such a large social network to collect these herp feeding data. So we had more than 1,300 observers and roughly just over 1.4 observations per person. And notably, only 82% of people only saw one record, which is of note. And then if we look at, when snakes are predators, the number drops down to 1.24 per person. And the skewed frequency distribution really highlights just how rare it is to see a snake feeding on something in the wild. And I do think this represents reality rather than somebody failing to upload the content. I think it's too interesting for somebody not to share something. So it is really quite rare to see a snake feeding in the wild, I think. And so I do want to point out that not all the photos are as lovely as this one depicting a vine snake eating a green snake. We do get some other ingestion states. So we get a lot of instances of animals regurgitating their prey, or their prey, while they're eating their prey, they get disturbed. And there's also photos being uploaded of road-killed animals with prey in their gut. And so these sorts of observations come from most people who are snake removers or keen herpers, and they are the ones in the previous graph, the 0.5% of people who have uploaded more than 10 predation records. Okay, so what do these records have to offer? Do they change our current understanding, and have we learned anything new? Are they just telling us more than the same? To do this, though, I needed to create a baseline for comparison. And I did this by conducting an extensive literature review, and I built a data set of all the feeding records pertaining to snakes in Southern Africa. And this data includes published scientific literature and gray literature, but it also, notably, it excludes captive feeding records and any sort of unsubstantiated secondary record. And one of the things that I found most striking was the difference in the accumulation rate between the two. On this graph, the gray bars represent the accumulation of dietary records from the literature in the last 140 years, and the blue bars represent the records that were collected from Facebook between 2015 and 2019. And so these records that we collected from Facebook essentially represent 27% of all known snake feeding records for Southern Africa. And so this represents one of the largest increases in snake feeding data ever observed. So another way to say this is that if we do a moving window analysis, there were no other periods that there was as much data collected as there was during the period when we were running this Facebook project. And the closest was between 2006 and 2010, when there were on average 139 records being published per year. And this is because this is when several museum specimen studies were taking place or were being published. And these comprehensive studies were conducted on entire clades like Samophines and Southern African Cobras. And it's important to recognize, though, how long it actually takes to collect museum data, right? So besides the actual dissection time, the data is the product of field excursions, specimen fixing, and realistically, they've been sitting on the shelf for the last 60 years. So it's not as instantaneous as the Facebook records are. So that was our first observation. Using Facebook in this manner resulted in a rapid accumulation of snake feeding data compared to historical approaches. Next, we wanted to know whether the approaches differed in how well they were identifying prey. And so just to back up a little bit, within each data set, assess the taxonomic level in which prey were identified to. And I found that the prey were identified to a finer taxonomic level using Facebook in all cases. So a larger proportion of Facebook records have prey that were identifiable to the level of species, as well as genus, family, and even order. So I think it's always important to point out now that a predator doesn't necessarily care about species delimitation, and taxonomy is a human construct. But I would argue that we miss a lot about the species biology, without species-specific information when you start aggregating and clumping prey. Okay. And so I can only speculate really why we might get these patterns. So many times museum-based studies have prey it's really hard to find any key diagnostic features. And it might be the case that all of our in situ feeding observations just really help to boost the ability to identify those prey. So I just want to go on to how we've further point out how we've been able to further identify prey more precisely. And so on this graph, I'm plotting each dot represents the niche breadth or the prey diversity within the diet of a snake species. And so what you can see is that the graphs are showing largely, the graph is showing significantly similar levels of prey diversity between the two approaches. And so what this is either saying is they're both sampling in a biased way, or in a way that is, let me back up. Our Facebook group isn't simply sampling the same thing over and over again. We are sampling a variety of prey. Or we're doing it in a way that is similarly biased to what previous methods have done. Okay. But let's look at the actual identity of these interactions. So let me just walk you through how I analyze these data. So to make comparisons of the species interactions more straightforward, I just took the records that we had species level identifications for. And then from there, what I did was I eliminated any duplicate interactions. These are non-redundant interactions within Facebook and non-redundant interactions within the literature data set. So then I took those interactions and I compared them to each other. And remarkably, what I found was only 9% of the species interactions are shared between the two data set. And notably, what that means is 74% of these non-redundant interactions that we detected on Facebook were novel. And so I'm just presenting here what's called a Cohen's Kappa coefficient. And essentially, this large negative value indicates that not only are these two approaches highly discordant, it means that they're doing so in a way that is non-random. Essentially, these approaches are differently suited for detecting certain interactions. And if we redo the analyses at each taxonomic grouping, we still find that Facebook has produced a number of novel interactions. And we similarly get a kappa value as negative as we did before. Okay. I just want to point out the species for which we have more than 5 novel interactions from Facebook. And it's worth pointing them out because they are all generalists with large geographic distributions. So for instance, the first species there, Burdon capensis, the brown house snake, is a small-bodied constrictor and basically eats anything from birds to mammals. The other members on the list include whip snakes and also cobras, which take the, if it fits in my mouth, I'll eat it. And so then it's not so surprising that we're still detecting new feeding interactions because these generalists do consume a diversity of prey, and the more sampling effort you expend, the more likely you are to detect more species in the diet. Okay. So what have we done with these data? What do we want to do with these data? So first of all, they've been really useful for describing just how incomplete our understanding of feeding interactions involving snakes has been. And it has been a way to fill this gap. With the brown house snake Burdon, I mentioned we have lots and lots of Facebook records accruing, and we've now supplemented them with museum data, and we're testing whether spatially mediated differences occur. So are urban snakes eating something different than non-urban snakes? With African pythons, we're exploring what it means to be able to take these jackpot meals, which is a huge shift from their diet of small birds and mammals in early life. With boomslang, we've been able to show that sexually dimorphic coloration does not result in any differences in diet between the sexes. With African cobras and Cape cobras, within the clade Naya specifically, we've been able to show that ophiophagy is ubiquitous and cannibalism has been found in at least 5 of these lineages, suggesting these prey choices may be an ancestral condition. And across the clade, we've learned that puff adders account for 27% of the snakes ingested, which is paired nicely with our observation that puff adders appear to be resistant to Cape cobra venom. And eventually, maybe sort of a pipe dream, we'd like to be able to compare functional groups across habitats and test for temporal differences in diet, but we do still need a bit more data to be able to address these questions. So I've only focused on the snake aspect today, but I want to point out how are we doing for time? I want to point out how we are also collecting the diet for what lizards are eating, what anurans are eating, and also what is eating all of those herps. And so this could eventually potentially inform topics such as interguild predation and functional redundancy within a community. So that's sort of the end of how using social media as a community science platform compares to historical approaches. But what about how it compares to dedicated community science platforms like iNaturalist, or through passive crowdsourcing of information by looking through Google Images? Similar approaches could probably be done through iSpot or HerpMapper or even Zooniverse. And for passive crowdsourcing, I just want to highlight this term called Ecology, which was coined by Yarrick et al. Last year. And it's this idea that people are already posting a lot of information to the internet and to social media. And there's this opportunity that we can just passively collect them from people. So it's not this active engagement with a community science type framework, but those data already exist. Can we utilize them? Okay, so for the iNaturalist portion, I utilized an already available project on iNaturalist called Interactions of Southern Africa. So it has, at the time, actually up to date number now, 32,000 observations in the project. And notably, it was created as a way to log interactions on iNaturalist, since there isn't a method that's formally incorporated into the platform. This project, it's a little bit clunky in that you have to log both interacting species, and then you have to upload them to the project from there. But it's a way to at least collect some of those observations. They're not just floating around. And it's important to note that this required a lot less time than the Facebook method. If any of you are familiar with iNaturalist, you'll know that the species are already identified for you, the GPS is already uploaded, and there's a query system that you can just download the CSV file. However, what we found, there weren't nearly as many snake feeding records. So I could only find 102 snake feeding observations. Going back to 2011 was the first. And interestingly, there was significant overlap with the species that were detected feeding on the 2 platforms. So the brown house snake was the most frequently detected species on both. And it's likely, as I mentioned before, due to their tolerance or even attraction to urban environments. And importantly, we detected species interactions using iNaturalist that weren't known from either Facebook or the literature. So there were 13 novel interactions that we detected. And then it's also worth noting that there was a degree of overlap between the 2 platforms in terms of exact duplicates. So these are photos that were the exact same predation event uploaded to both platforms. So next I want to talk about the comparison to the purely passive crowdsourcing approach. At the time when I did this, I didn't use any sort of highly automated approach. Literally just did a search, various search terms for the scientific species, because it's important to remember there's no grouping or there's no project on Google Images that you can look through. And so I restricted my searches to just 4 target species, which were the brown house snake, Python natalensis, the African python, Dyspholidus typus boomslang, and Nyanivia cape cobra. And I did this because they were the 4 most popular species on the Facebook group for data that we collected. And so I used the search terms for those species and then various feeding and predation search terms to go along with it. Okay, so this graph really emphasizes the difference in amount of records that we were able to collect across the platforms. And so for the Google image searches, it really didn't do nearly as well as Facebook. I mean, iNaturalist didn't either, but Google did outperform iNaturalist to some degree. And so what I did next was I took these observations and placed them into broad prey types just to see if there were any noticeable trends or biases in the prey that was detected by these methods. So what I'm presenting here, on the left is the original proportion, on the left is the number of observations for the brown house snake. And then on the right is the proportion of feeding observations where the prey belongs to one of the below prey types. And so for the brown house snake, Facebook and iNaturalist were more comparable than Facebook and Google. Although with Google we detected, we did have some detections of like the infrequent prey types like frogs and also birds. But if we then look at African python or Cape cobra, there are some very noticeable differences in the proportion of prey detected for these. If we look at the graph for African python, you can see this. First of all, there was a failure to detect any birds in their diet. And secondly, these sources seem to be heavily skewed towards very large prey type like antelope. And so this is sort of speculative, but it's these meals that are spectacular to watch, photograph, and share. And the length of these processes means it could be detected more easily. And a similar story might be true for Cape cobra. So we see from the data on Google that there's this huge bias towards snakes in the diet. Moreover, all of these snakes that were recorded were either puff adders or mole snakes, both of which are very large animals. So this could suggest something about large meals being more easy to photograph. But notably, these are also very headline worthy. I mean, look at that photograph. It's quite a photogenic event. So just to summarize the findings from the Google Images portion, roughly 20% of these predation event images also appeared on Facebook. It's just important to note at this point that at least when we did this back in 2019, groups and their media weren't being indexed by Google, but that has now changed. So if you were to rerun the search now, there is a chance that number would even increase from this point. But at the time, it was just people were sharing them to Facebook, and they were also sharing them to some other website or something, or they were in the headline kind of thing. Importantly, there were no novel species interactions detected using this method. And most importantly, I think, is that this was a very time-consuming method, and I think it was quite prone to errors because you often get a lot of images from the same event, but you can't classify them necessarily with any software as the same event because they're different poses, but they all relate to one predation event. And so for that reason, I don't think it was as useful as, say, iNaturalist or Facebook. And the other thing is that the location wasn't always clear, which makes for some uncertainty around the ID and any sort of spatial analyses that you want to go forward with afterwards. And so ultimately, while an approach like Google Images might be useful for some taxa or some questions, the results in our case suggest that it wasn't the most fruitful approach and not the best way to study reptile feeding ecology, at least in Southern Africa. And just to point out that even though iNaturalist had many fewer records, there were novel records detected at least, and novel interactions detected at least. And I think that it could be really powerful in regions that have a lot more data. So regions like the United States, for instance, which have over 900,000 observations of reptiles. And so, you know, there's a mind-blowing amount of data for some of these species. The Western fence lizard has almost 60,000 observations. I mean, that's incredible. There's incredible scope to address questions related to reptile ecology with that much data. Even in the more data-sparse regions like South Africa, which has around 32,000 observations of reptiles, or Canada, which has just over 50,000 observations, there might be opportunities to address species-specific ecological questions using some of the species that have more data. Okay. So there are several challenges I've sort of mentioned throughout, but I just want to highlight here regarding Facebook or social media in general when it comes to studying reptile ecology. So as I've alluded to, all of our data collection was done manually. The Cambridge Analytica data breach back in April of 2018 meant that we couldn't use any sort of API function. So without doing illegal stuff, we had to just collect all the data manually rather than scraping it. And so eventually maybe machine learning algorithms may have a place and there might be enough data, but at this point, in terms of any sort of machine learning detection of prey that snakes in Southern Africa are eating, it's just there's not enough data to train those algorithms at this point. And then I want to point out how content is never guaranteed. So at the user level, even if people share a post, they might delete it or change the viewing settings. So it's never guaranteed to be there into the future. And similarly, I feel like it's every other day that Facebook's in the media. We just don't know how long certain social media platforms will last. And so it's just worth considering how data are being stored and how they're being captured for your study of interest. And so unlike iNaturalist, expansion into other regions using Facebook, it does require the researcher to have their own network, their own collaborations set up, because you can't draw on the power of just somebody logging on and IDing things. It just, being on Facebook or these social media platforms, it just is a little bit difficult to expand beyond your network. And again, unlike iNaturalist, where it's linked into the taxonomic, the taxonomy is updated regularly based on some all higher being. I don't know who decides it, but at least there is a way to keep track of when taxonomy is changing. When you do these methods, you have to decide what species you think it is and what its current name is, and it just takes a lot of effort to keep all of your taxonomy up to date. And importantly, going into the future, it's important to keep those photos safe, because if somebody wants to reference it in the future and it's not there, then you're not really sure if that prey item was that species. Okay. And so data completeness. So especially for platforms with limited enforcement of what's inputted, it's always challenging to make sure everyone is uploading their location. And so these can just, there's certain data completeness problems that you have to be aware of when you're going forward with these projects. And finally, it's worth considering the role of all of the community scientists in your project. And I refer you to this paper that came out in Tree last year that just talks about how do we actually address and include the community scientists in our project, because really without them, we wouldn't have the data. So it's worth having journals change who is given authorship as we go forward with community science. Okay. So just to quickly summarize, I hope I've convinced you that Facebook can be a viable community science platform, which works best for events that people are already sharing photos of. And here for my project, I've showed how it resulted in a faster collection of data at a finer resolution, which has informed us of many new feeding interactions involving African snakes. And while this method is powerful, the data, both from the literature and iNaturalist, has really emphasized that it's not a replacement, but rather it should be used to supplement current strategies, especially if we wish to build a comprehensive and robust data set. And ultimately, going back to natural history, it's important to consider the natural history of an organism to achieve a better understanding of it and to recognize just how many assumptions we are making when we don't have a complete understanding of their natural history. And with that, I just want to thank Brian Maritz, who has been my collaborator and partner in all of this, and to thank all of the contributors to the Predation Records group. As I said, we wouldn't have this project if people didn't submit photos. And thank the group admins and moderators who helped maintain order and civility. And finally, thank you to everyone who's contributed their expertise and assisted in identifying predators and prey for us. And I'm happy to take some questions if there's time.
Amanda, do you want to go first?
Sure, thanks, Julia. And thank you, Robin. That was really, really interesting. I was quite struck by the lack of overlap in the taxonomic observations between Facebook and the museum specimens. And I was wondering if you had any sort of ideas as to like what those characteristics might be that you're not capturing in one versus the other, or what that looks like.
Yeah. So I haven't looked into it too deeply, and it is something that's been on the back of my mind, just to see is it frogs that we're missing, because that's the idea, is that maybe things like frogs, we know from the museum specimens they are breaking down. It's really hard to see them in the gut, and so we can say that it's an anurin, but we can never say what species it is. But roughly when I looked, it's that we're just adding it. We are adding new taxa in, and I think in some ways it's because the number of species is increasing. But beyond that, I do have to look a bit closer to see if it's a certain prey group that's being added all the time. But my gut feeling is we just are able to pick up a lot more when we can see the animal rather than just paste inside.
Probably. Some of those museum specimen samples are quite degraded.
Yeah,
Jane,
exactly.
do you have a question? There you are.
Hi, really great talk. I was wondering, given the challenges with scaling up like the Facebook approach, have you experimented or thought about like just encouraging those people to submit to iNaturalist, because then you get around a lot of the data completeness and archiving issues and permit?
Yeah, I mean, it's definitely something I've considered. It's worked to some degree with the distribution work. There's a team of dedicated people who always say, please upload to the virtual museum, please upload. It's sort of become, they will upload for you. And the challenge that I see is, so instead of me sharing it to the group or somebody sharing it to the, we always say, please share it to our group to get them to encourage. And even with on Facebook, it's really hard to get them to even do that much. And so it is a potential to get more data, but I'm skeptical. Yeah.
Great, thanks.
Thanks.
I think Stephanie Wilson has her hands up. Sorry, I'm trying to figure out all the hands. Okay.
Thanks, Julia. And thank you, Robin. That was super interesting. I loved seeing all of those amazing photos.
Yeah.
I was wondering, and maybe this isn't possible where you are, but I was wondering if you had an opportunity to look at any sort of conservation data center data, because I know that's a big thing here in Canada, is submitting to the CDCs. So just wondering if you were able to look at that data source as well.
I guess the closest would be the virtual museum that we have. I haven't looked into it too closely. And the other thing is that a lot of the occurrence data was going initially through iSpot, and so it was also being pushed through that avenue, and then that was transitioned to iNaturalist. And so there isn't besides the virtual museum, which I haven't looked into too close, there really has been this push to iNaturalist, which I've already collected those data from.
Awesome. Yeah, we have some more, anyway, some other data repositories here in Canada that I think maybe Stephanie was talking about. Pam, do you have a question?
Yeah, I do. Thank you, Robin. You spoke to this, I think later near the end of the talk, but it sort of occurred to me throughout trying to address systematic biases in the data set. So could you expand on that a bit? I guess I had a few pop into my head as to what might be biased within Facebook. And I'm also curious if you have any thoughts going forward about addressing any of those systematic biases.
Yeah, so we've always been concerned that, for instance, with our work on Naya and just seeing the extent of ophiophagy, that it might be that, well, if somebody sees a photo or if somebody sees a snake eating a snake, like it's really like, oh, I must share this. But I would argue that if anytime you see a snake eating, you're probably going to share a photo. And so in that case, it might be that maybe a snake takes a bit longer to go down because it just is a larger prey item. But at the same time, we got records, this animal on this last slide here is a vine snake. We have a record where it was chasing a little tiny rain frog and gobbled it up in 30 seconds. And so even though there might be biases towards some large prey items, we are also getting prey items added that have very small handling time. Yeah, so definitely there is a concern about handling time, more likely to see it if it takes longer to go down. And then, I mean, people are taking photos around their house, so maybe there's some sort of bias in that regard. The prey around the house is not representative of the rest of the habitat. And so, yeah, I mean, it's worth doing some sort of systematic analysis of museum specimens are biased in their own way, but at least comparing what does it look like in terms of the size of prey that are consumed on the data set from Facebook versus the museum and things like that. So I think size is probably the biggest issue. And I mean, the other biases is we're never going to get these underground burrowing species. We're never going to see what they're eating. And so that's just something we have to accept. We'll never get some of these typhlopids that they live in termite mounds and only eat termites. We're never going to see those. Yeah. I'm curious what your thoughts are.
Oh, okay. Well, James has some thoughts.
I have a hand.
Okay.
So building on that kind of exact statement and tying back to what Amanda was asking in like the first one, do you think that like that might play into the discrepancy that we see in like the literature and museum specimens versus like what's being captured online? Because, you know, when you go out on like a museum expedition to catch stuff, it's not like you're hunting around downtown Cape Town, like you're going off into the feld kind of thing. So do you think that like the natural biases within like scientific trips versus what people see in their backyards
for sure
might be why you see those kind of separation of different taxa?
Yeah, definitely. Yeah. And I mean, if you've ever spent any time in a museum collection and say you can only dissect 20, you grab the ones that feel like they have something in it. And so you are going to miss a fair amount in that way. And also if you're out collecting, there's a chance you stumble across something that's bigger because it's a bit slower. And so there are several ways that you could see these biases popping in. Yeah.
I think we don't have any more questions. I just had a quick one. For the literature search, were you able to look at like natural history records? Because I would say before there was Facebook, there were like natural history notes, and now Facebook has kind of taken over from that, which I'm glad that you've like jumped on. But were you able to include those in the literature search that you did?
Yeah, like all of the, I mean, it's difficult as it gets beyond like 1950s, but I tried to do my best of gathering just all of those anecdotal reports everywhere. Yeah, all of the
Herp review is hard because it's not indexed, so
it's not very searchable. No,
Yeah.
so I definitely went in and did targeted searches through some of the known society bulletins, including the herp ones and also the bird ones, and so it was definitely a targeted approach as well.
Yeah, that is really hard for the herp literature searches, I think, especially for interaction data.
Yeah, and then it becomes difficult because, you know, somebody publishes something in the 60s and you realize they're probably talking about the same thing in the 80s, but they don't clearly say it's the same specimen. So it takes a lot of hunting.
That's really difficult. So I think everyone's questions are done. So anyway, I just want to say big thanks. The interactions you're capturing are incredible. It's stuff that I wish I have seen, but I'm glad someone has and they've taken a photo because it's so important for us to know about. So anyway, just let's all give Robin another big round of applause.
Thank you, everyone.
Everyone.